This is a representation of double-stranded DNA, like the double helix shown on the first floor sign. The underlined letters are particular components of a gene. The right-angled arrow is the transcription start site, or TSS. That's where RNA polymerase will bind to this DNA molecule and start producing an RNA transcript. The TATAAA sequence is the consensus sequence for TATA binding protein, which is a transcription factor that will bind to the DNA at that sequence and then help bring RNA polymerase in and place it at the transcription start site. The other underlined series of letters here, AATAAA, we're going to see in the next floor sign, which is an important sequence, the polyadenylation signal sequence, which is important for transcription. Here on the second floor, we're dealing with the second step in the central dogma of biology. First floor, DNA. DNA encodes a single-stranded nucleic acid, RNA. And in this case, the underlined letters are important both for RNA processing, which is a step that happens in eukaryotes, and also for the next step, which is going to be translation, which we'll talk about on the third floor. So the underlined letters in this messenger RNA molecule, the primary transcript includes what's going to be the translation start site. That's the start codon. We're going to talk about that on the third floor. The GU, A, and AG are consensus splice sites. Again, in eukaryotes only, this is important for the process of splicing, which is going to remove everything in the messenger RNA molecule between the GU and the AG. So that whole bit is going to be cut out through processing, and that's why we see that it's no longer present down here in the processed transcript after splicing out of introns, which is what this is, occurs. Other letters here include what's going eventually to be, on the third floor, the stop codon for translation. And again, we have the polyadenylation signal sequence, which we saw on the first floor. And that is still here in the process transcript. And the position of that sequence directs a new enzyme, poly-A polymerase, to add untemplated, or essentially random, strings of A's to the end of a processed transcript. So in the bottom, in other words, we start with the initial transcript at the stop. After RNA processing, three things have happened. First, a new nucleotide has been added to the 5' end of the transcript, a 7-methylguanine. The start codon for translation is present. Everything in between the start of the transcript and the start codon is the 5' untranslated region, or 5' UTR. That's because this is part of the RNA molecule, but it's not going to encode protein. The bit of the RNA molecule that encodes protein is what exists between the start codon and the stop codon. So that's the coding region of the messenger RNA molecule. Then everything between the stop codon and the end of the transcript is the 3' end of the transcript. That's the 3' untranslated region, doesn't code for protein. And again, we see that poly-A signal sequence, which directs, after about 25 or so nucleotides, the addition of the 3' poly-A tail. And finally, here on the third floor, we see the product of translation. After the ribosome binds that 5' 7-methylguanosine cap, scans to find that AUG start codon, then the ribosome produces a protein, in this case a series of amino acids, all covalently bonded to each other through this backbone. And the side chains of these different amino acids indicate their identities. We have 20 different amino acids that are naturally occurring, and this is a set of those amino acids. So one question then is, what is this protein that the gene on the first floor and the messenger RNA on the second floor encodes? If you look up the structures of the amino acids, the first amino acid in a protein, which is the amino terminus, the N terminus of the protein, with the carboxy or C terminus down here on the left side. So there's the N terminus and the C terminus. And that's the direction we read amino acid sequence from N to C, left to right in this case. Every protein starts with a methionine. Then we have a tyrosine, whose single letter code is Y. Then we have serine, cysteine, isoleucine, glutamic acid, or E in single letter code, asparagine, N, another cysteine, and another, that's a glutamic acid. Did I say aspartic acid? These are glutamic acids, E. So, what does this spell? My science. Thanks for watching.